change the slide. I store my my slides, my old slides, at this box, which is a humidity chamber. It's basically a few stands for the slides and water with cotton. Let me clean the slide from the water. Here you can see lots of filamentous algae. It is the green stringy stuff. And I don't know if you can see, but you should be able to see little things moving around. Now, since this is an old sample, an old slide, the water has started evaporating, even though it was in a humidity chamber. But they can hold up for a very long time, especially if you replenish the water. Now, for some reason, my chat integration has stopped working. I don't know why. Let me fix that very quickly. Okay, shut. Okay, there we go. Okay. Now, I'm not seeing currently anything wildly interesting, but that may change once we go up in magnification. This is, by the way, 40 times magnification. Now let's go to 100 times, and here you can see lots more detail. And again, the algae. I think there are a few diatoms attached on the filamentous algae, on the string here in the middle of the screen. You can see them. And there are a few microbes running around. Oh, I think there, let me change magnification again. Now, those I think are in the middle of conjugation, which is basically sexual reproduction in ciliates. It is a process where two individuals exchange genetic material, and essentially they become their own daughters. They're essentially daughter cells. And in every species there are like two types of reproductive I would say reproductive types, type 1 and type 2, and depending on which type each microbe belongs to, it can merge and begin conjugation with the other type. Now, I don't know either the species of those ciliates or the genus, um, but it's awesome to see them in the middle of conjugation. Now, here are the diatoms I was saying before. You can very clearly see them. They're attached to the algae, which probably helps them get more light, because generally algae can grow higher than the diatoms, which then get to the surface of the water, and then getting more light, and also more carbon dioxide, which either diffuses into the water or rises from the bottom of from the sediment, where there are generally more microbes. Now, let me see if you can see them. There should be some strings right in the middle of the screen now. A single string can easily be discerned. Those are colonial bacteria, I think, and they form up strings like this. I don't know why. I haven't read anything about them. Now, there is this structure in the middle of the screen right now, which you can clearly see. I think this is a hair. I'm not sure though, but judging by the scale-like structures that are on the surface, I'm guessing that it is indeed a hair, a string of hair. Oh, here are more microbes. Again, here are two ciliates in the middle of conjugation. A little more about the conjugation. The ciliates form merge by merging their how's it called? Their cytoplasm with a cell membrane merging, and from that opening they exchange genetic material, so they can increase the diversity among the population, because after a certain amount of cell divisions, the cells can accumulate genetic damage by mutations that naturally occur in the genome, and the mutations can lead to various evolutionary disadvantages. And conjugation basically is the process where with sexual reproduction they manage to mitigate the damage, the genetic change. Obviously, good mutations stay with them, because not only stay but propagate through through the generations, because obviously they offer an advantage to the ciliates. Now, those are quite small ciliates, and I have an identification book right here. Let me see if I can find anything related to them. They seem to have some of them. They seem to have some spherical structures, so at least circular, as I can see, as far as I can tell, that are probably food vacuoles. Basically the food that they have eaten is stored in there to be digested. And here, let me focus it. Right in the middle of the screen there should be a string, a wavy string. That is a single bacterium, a spirochete. They are spiral, it is a single organism, not like the strings that we saw before. And some of them can cause some diseases to the humans. And I cannot remember which diseases. And they essentially move by spinning their flagellum, which is different, by the way, from the flagellum. The bacterial flagellum is basically a spinning motor which spins a string-like structure and propagates the bacterium. The flagellum is a structure made from microtubules, and doesn't only spin, it can move in more advanced ways. Now, regarding the ciliates, I haven't found something resembling them. And I don't think they are either spirotric ciliates or hypotric ciliates. And I'm really not sure where they belong. Anyways, let's see what else this slide has to offer. You can also see some smaller ciliates, but I cannot discern any details to tell you anything about them. There are and there should be a lot of debris around the slide, because debris generally contain food for the microorganisms, and also provide something to latch on for some of them, like for example stentors, or maybe or microorganisms that are multicellular, like the rotifers, some rotifers that are sessile. Oh, I think I can see an amoeba. Right next to that diatom, you can see a small and very faint structure. That I think is an amoeba, and I think I can say what type of amoeba. I think there is an example of that here. It is for sure a naked amoeba, and it could be let me see again. Perhaps either Platyamoeba or Vanella. Um, and I think I'm leaning. I'm not sure. It's probably some species related to them. There are several of them. I can see at least 3. Let me see if I can show them to you. The amoeba has moved, as you can see, is going right under the hair. And there is one other amoeba right in the center of the screen now. What seems like is debris with like a flap. That one has eaten a very small diatom. You can just barely see it. And this one also, right in the center of the screen now. You can just about discern it. And also you can just about tell that it's moving. Now let's open the diaphragm. Now, generally in most of the samples, it's very easy to find diatoms. Diatoms are literally everywhere in water ponds, and in generally most water samples, you are going to find diatoms. There are loads of species, and you can see several of them here. The big one in the middle, it's quite a small one actually, compared to some other ones. And those smaller ones. And you'd think that diatoms which are made of, which have silica shells, they wouldn't move as much, or at least they would move very slowly. Now here I don't see any moving diatom, but they can move pretty fast compared to what you would think, at least. Compared to some other organisms like, for example, ciliates, most ciliates at least, they move pretty slowly. and but for example, compared to some amoeba, they they can move quite fast. For example, those amoeba, this one in the middle of the screen, it doesn't, you can see it very well, but I can assure you there is one. Let me close again the diaphragm. Now you can see it quite clearly. This moves quite slowly compared to many diatoms. I'll see if I can find a moving one to show you. Now, here, for example, is a ciliate conference. I don't know what's happening, but I'm guessing in this area there are a lot of bacteria, so those ciliates can easily obtain nutrients to reproduce, to grow and reproduce. Now, that diatom is not moving on its own. Oh, I lost it. Here it is again. It is. It has unlatched from the either the slide or the cover slip, and now it's getting dragged by the currents made by the cilia on the ciliates, obviously. Here are two ciliates again in conjugation. Here is another species of diatom. I haven't seen any of those diatoms that are latched on the filamentous algae moving. Oh, here is an amoeba. This is another species of amoeba from a different category. It has seemed to eaten a few diatoms. You can see one right in the end of the pseudopodium, and also you can very clearly see the cytoplasm moving inside the cell. Now, amoeba are very flexible, and their form can easily change, and they move by extensions of their cytoplasm and cell membrane, which forms those structures. You can see those that look like appendages and are called pseudopodia, and also by those they eat other organisms or anything that can be food to them. Now, here is another species of ciliate. Oh, it's gone. Now, this amoeba seems to be latched onto the slide. I don't know why, honestly. And is probably looking for other organisms that are passing by to latch them and engulf them with cytoplasm and form cyst around the organism, and then insert digestive enzymes inside the cyst, and then obviously digest them, and then make excretions. Now let's move on. We'll return to the amoeba later, probably. Although I would like to check out the... Oh, I think this organism, let me see. Shame. This organism seems to, the ciliate seems to have very long cilia, and I think I have an example of a similar one in the identification book. Also, there were some amoeba above that I'm going to look at next, but let me check. Now, I think this could be part of the genus Euronima. Or not. It doesn't have the shape that the identification book specifies. Oh, what was that? This, I don't think you saw it. There was something moving, and I think that was flagellate. No, unfortunately, there doesn't seem to be any specific example of the organism that was there, from the ciliate, I mean. And here are a few amoeba. Let me check a thing first. Okay. So these two are probably the same species of amoeba. If I'm correct, they seem to have similar patterns. I don't know if they're similar to of the same species, or at least genus of the previous one, but it's very likely, and they could be looking for a place to latch on and start eating. Here is another amoeba. Let me see if you can see it. Now you can see it quite clearly. Now we are on the edge of the water, and there doesn't seem to be anything very specifically exciting. Now here are a lot of dead or at least dying organisms. Now let me add a little bit of water to replenish the water in the slide, because it has started being too little. Now there is a very good way to add water to a slide without removing the cover slip, or putting water onto the cover slip. You dip the tweezers onto the jar of water, and you close them inside of the water, inside the jar. Now this causes the water, some water to right now you can see that just fell onto the slide. This causes the water which wants to have the minimum surface area to latch on to to stay on the tweezers, on the closed tweezers, and when you open the tweezers, the water loses its some of its surface area that it has been on, and it causes it to drip and go right into the slide. Now the slide is quite full of water. Let me clean the rest. There we go. And let's put it back in. Okay, so we're, oh, I accidentally reduced the light intensity. Okay, now we're back again on the slide, and right now I don't know where we are because I moved the cover slip accidentally. Here's a bunch of dead diatoms. You can see their empty shells. Let me it's a bit blurry on the camera. Okay, now it's better. The shells, as they're made of silica, they stay well after the death of the diatom. Here also you can see the shells of some bigger diatoms. You can also see that the middle one, the oval shell, is broken, which could mean that it was eaten before it died. Now let me see if I can find anything else, and then we'll probably move on to the next specimen. I think this is the same species of ciliate as the previous one, the one that I thought was the I don't remember its name. Um, oh, here is another mass of ciliates. Um, Now, this slide I am going to keep after the stream and keep it in the humidity chamber. I have two of those, so for now at least I can store at least seven slides. Here is again those ciliates in the middle of conjugation. Now you can see them better because they're not moving, which is quite good. Now here is again a diatom that has I think it just latched on the slide because it was moving in a way that diatoms generally do not move. Now here the concentration of those ciliates is surprising, really. I have seen in that many, in that large of concentration, only flagellates of probably the genus. Let me see it again. Sometimes I cannot remember names. Oh, Chilomonas. That's the one. And that is quite a large concentration, quite a high concentration. I think this green thing in the middle could be a dead Euglena. Euglenas are flagellates that are, most of them at least, are photosynthetic. Right now, in this slide, obviously we haven't found one. Again, lots of diatoms. Generally, you'll find diatoms either latched on filamentous algae or on the top of the slide or on the bottom of the cover slip. This amoeba is currently just a diatom, the golden thing that you can see. It just engulfed it. Oh, that is something I have never seen. This is another amoeba, and let me see, because I can remember correctly. The amoeba is the star like the cross like thing in the middle of the screen. You can see right now. But let me see, because I think there is an example of such an amoeba here in the book. Um, oh, it is. It is probably a vanilla. The vanilla when it's not moving, but this one is moving. Like, see the sketch. This one. And I think all of those flat amoeba, the small ones, the ones that you can't see easily, are vanilla, of the vanilla genus. The vanilla genus seems to have, at least according to the book, seems to have way larger pseudopodia when apparently not moving. It is slightly moving though. And here is vanilla, probably at least, right in the middle of the screen now. Oh, let me focus. Let me close the condenser a bit more. There is the vanilla apparently, and there is the sessile form, and obviously around there are, at least I can see currents of bacteria that are not unfortunately the program that I'm using to transmit data from the phone's camera to the computer doesn't have that much resolution, so it's having a problem discerning some things. But here there are obviously lots of ciliates moving around, and there I can just about see some bacteria moving around from probably the currents of that are created by the ciliates. Very awesome though that I can see two forms of the same species. Let me open the condenser again. That should be good. Okay. Oh, here's another amoeba. Right in the middle of the screen, latched onto the hair. Let me close again the condenser so you can see it better. Now we're talking. You can just about see the cytoplasm moving. I can see it a little bit better. And you can also see the amoeba extending the pseudopodia towards the right side of the screen. Also, let me see chat right now because there have been some messages that I haven't seen. Oh, okay. Okay, let's continue. I'll be honest, I didn't really expect this slide to be that rage. I was expecting less results, less impressive results. but I'm quite happy with the stream for now. And we're just about to close an hour, so let me check real quick. Let me change magnification again. Now we're up to 100, down to 100. Let me open the condenser. Oh, here is a flagellate. I think you can see the flagellum. Let me see. You just about can in some frames, but this is a flagellate. Again, I'm not sure about the species, but I think this particular species, I have seen it again in slides from another sample. Let me see if I can show you the flagellum. You can just about see it now. Yep, so the flagellates move in a very particular way, which they get dragged by their flagellum. They use it essentially as to move for motility, for mobility, yes, and they are a bit more flexible and more palpable. Is that the word? I think it is the word. Than the ciliates in general. Here it is again. You can see it moving a bit sideways due to the flagellum. The flagellum is obviously in the front because they get dragged. This one is not photosynthetic, obviously, because there are no green stuff, which are basically the photosynthetic come on, I'm forgetting words. Until I remember the word, let me check for the species. So the species that seems to represent the flagellate is either Rhabdomonas or more likely Paranima. Here it says that the Paranima is quite small, but way bigger than Rhabdomonas. So I'm not sure, but I'm going to say that it is more likely Paranima. I think it is more common too. I think we've lost it now, but let's see if I can find it again. Here again we're at magnification 100. Oh, here it is. On the lower magnification I can do this, which is basically it is called Oblique Illumination, and it adds 3D effect with shadows. I'm not sure if you can see it from from the stream. No, I don't think so. The resolution is a bit too low for my liking, unfortunately. I'll see how I can fix that in future streams. But yeah. Yeah, the word that I was looking for is chloroplasts. Yeah. The chloroplasts contain chlorophyll, which is the green pigment, and it is basically the base of photosynthesis. It is the molecule that does the photosynthesis and creates sugars from essentially carbon dioxide, CO2, and light and water. And the products are basically oxygen, which is what all non-photosynthetic organisms breathe. Even the microbes here, they breathe through their cell membranes. The oxygen diffuses, and also the process of photosynthesis produces sugars, which are consumed by the plants, and in their turn, they're consumed by other organisms. Either they are microorganisms, like, for example, ciliates or amoeba, or from larger organisms that could be from plant-eating mammals or insects or whatever, or even small animals, microanimals, like rotifers or copepods and small crustaceans. Now, right now, I'm not seeing anything particularly interesting. Either that there is a bubble again, and this bubble is a result of the production of the oxygen production of the algae and the diatoms, and also I think it is a remnant from the water drying from the slide previously. Sometimes when there is enough algae around and there is no larger bubbles that the oxygen can diffuse into, you can see the bubbles forming from the oxygen bubbles forming. But now I don't think we're going to see that, because the algae, the large amount of algae is near to the gas bubble, the air bubble here, and all the oxygen is going pretty much there. Again, here, now I close the condenser so you can see a bit more. There are lots of diatoms and lots of ciliates roaming around. But I don't think this slide has much more to offer, for now at least. And again, here you can see the border of the water and the air. Here is the dried stuff, and here is the end of the slide, as you can see, the end of the cover slip. Let me refill the slide with water. And then we'll go to the next slide, the next sample. Generally, when you change water from the slides, when you do some precise things, like for example, what I'm doing now, it's generally best to keep the sample, the slide steady. For example, I have contact lenses case, an old one that I'm not using. So now I'm going to store it in one of the humidity chambers, and I'm going to make the next sample, the next slide. Now, those vials contain water from a large water tank that I have with a huge sample of water from a small lake. Now this, I think it's part of the sediment, and I would like to observe this one, which is contains biofilm that formed on top of the water, on the water surface. Now, let me get a slide. Slide here. Let me get the water out of here. I don't think we will be needing this anymore. Plastic pipette, transfer pipette. Those are very handy because you can transfer water in very small amounts. But what I'm interested in most is the biofilm, which is in a few large pieces. There we go, just took one out. There we go. Now let me put a little bit of water for the cover slip to rest on. You don't need a large amount, the slide has to be very thin, because otherwise you cannot use the full capacity of the higher magnifications, and also specimens like fast moving ciliates or generally microorganisms can move around and get out of focus very easily, and the smaller the thinner the slide is, the thinner the sample is, the more difficult is for them to roam around and get out of focus, so you can observe them more easily. A solution that I've read about is putting small amounts of Vaseline in the corners of the cover slip and then putting it onto the slide, so you can essentially press the cover slip the right amount, exact right amount, so the microorganism cannot move in the vertical axis, only the two horizontal axes. Now let me adjust a bit the form on top of the, there we go. Now I'm already seeing lots of stuff. Let's zoom out again. see if I can clean the condenser. Set the... Oh no, it is part of the focusing. There we go. So that is the new slide. For some reason it is quite dirty. Let me see. Oh. Oh, okay. Now it's good. Oh, so that what you're seeing now move around is was one. Now there are quite a few. Now you can see here that the water has leaked out of the slide. Larvae of small copypods, not small, copypods in general, and they're quite motile. And also I can see a large amount of single-celled organisms, which I don't know yet the species or the genus, or again, even if they're ciliates or flagellates. But here is the main part of the... Let me open the condenser. Here we go. The main piece of the biofilm. The biofilm could be either fungus, which has grown on top of inside the water and has floated up, or bacteria, which have obviously formed large colonies, or both. One that I had found was both and was quite impressive. Let me see if I can... Now this is dark field. I think it looks way better than bright field in lower magnifications. Here's another piece, and you can see a very large amount of microorganisms moving around, and a few frame drops, but oh well. For now at least, I can't do anything about it. Now let's go up in magnification and let's see what this slide has to offer. Let me close a bit the filter holder. Okay, now you can see, first of all, those ciliates that are not moving that much. Let me open the filter holder. Oh, they move. No, here they are. I do not recognize them, neither them nor the small ones. But I think it is a species, or at least the large ones I'm talking about, something that I haven't seen. And this one is the same species as this one, I think. You can see the large structure in the middle, the spherical one. And as far as I can tell, and this one, as far as I can tell, the smaller ones could be, are probably ciliates, and there are many different species of them. There could be a species of flagellates between them, but here is the biofilm. You can see lots of bacterial strings. And also, I don't know if you can see them, but there are some spirochetes among them. They're quite small though. And here is a gas bubble. It's not an oxygen bubble. There are not many photosynthetic, or even at all, there are not at all photosynthetic organisms in this slide. Here is a spirochete. Again, you can see it very clearly. Yep, this gas bubble got trapped in. Here you can see a bit more because the contrast is a bit better. You can see the bacterial strings, and I think there is a fungus in here. I'm not quite sure. Here is the end of the bubble, and again we're in the. Oh, here is a flagellate. This I'm sure is a flagellate that I haven't seen again. Again, let me check the book for more information, because I think there might be a species that represents that. Did we lose it? I think we lost it. Let me see if I can find it, because I think I found one that might represent the one that we found. Oh, here it is. So based on the shape, I think. Oh, that's. Is it a ciliate or flagellate? No, this is a flagellate. The ciliate, sorry. This is not a flagellate. And if it was a flagellate, I would have an idea of what it might be. But it is a ciliate. I can see the cilia in the smaller part, in the short, the more, I guess you could say sharp end or pointy end. There are clearly moving cilia. I don't think you can see it. You can just about see it sometimes. The movement. It's quite subtle though. I don't think I've seen anything resembling that. And I don't think there is something like that in the book. Nope, unfortunately. I'll try to research about them to make a little search and see what they are, the species of basically pretty much at least most of the microorganisms that are here. And I am recording this, so I'll be able to see the footage. Now this small one, I think this is a flagellate based on the movement. It's quite junky. Let me bring up the tab here. I think it went that way. No, that is something again I haven't seen. This is a ciliate, definitely, but it has something like an appendage on one end. Let me close a bit the condenser so you can see it better. And see if I can see any flagellums or anything like that. There is definitely a structure. I can see it sometimes. This small one that I'm following. But there isn't much that I can discern, unfortunately. No, oh, that was a larva of a copepod. Here is more of them. Those are flagellates, definitely. At least based on their movement. And here is a ciliate, small one again. Let me open again the condenser. Oh, what is that? This is the spirochete that has latched onto the flagellate. I'm not quite sure. And I'm not sure if the spirochete is trying to infect it or something like that. You can see the spirochetes here. Here's one, you can see clearly. Interesting, very interesting. Well, we have closed 1 hour and about 20 minutes streaming. Let me get here and see what's going on around the concentrated bacteria. This is probably a bacterial colony, which ciliates or flagellates, I don't know honestly. There could be both. They could be both. Yeah, I'm not sure what's happening here. It's probably a bacterial colony, a spirochete colony. Now this is another ciliate roaming around. It's from one of the species that we saw earlier. There is a large structure. It could be either a food vacuole, but it's quite empty, honestly, or it could be an oral opening. Let me see what's happening on Discord. Okay. That could be either of those, or it could be a contractile vacuole, which I don't think so because it's not pulsating. The contractile vacuole is basically a cyst that forms, and essentially it's used by the ciliate to pump water out of the organism because due to the osmotic pressure, which is essentially the need for the nutrients and other stuff to be on equal concentrations on everywhere. Water wants to dissolve all the things inside the cell and wants to rush in to lower the concentration to make it similar or same or identical to the outside concentration of molecules. And essentially the contractile vacuole is a mechanism to counteract that. Now here is a very fast spinning spirochete. Two of them actually. The one is bigger. They move by spinning their flagellum and spinning and essentially corkscrewing through the water. Now let's go down in magnification again. I would like to go and find a larva that's not quite moving. Here is one. Let me find it again. The thing is that since the the water border outside the slide, outside the cover slip, is quite large, they can roam around outside the cover slip, which is probably, holy shit, more comfortable for them. It's not... Oh, what's happening here? It's not very good for observing them, though. Now, this large bunch of ciliates, I really don't know what's happening here. I can see lots of bacteria, and the bacteria could be consuming the ciliates. Really, that's the only explanation I can give. Because at least I can see the bacteria. You can see them also. They form a ball, which at least a spherical structure, which the ciliates stick on, and they slowly go towards the center. I'm not sure though. Again, but the concentration here is another one, smaller. I think the ciliates are getting either stuck or being consumed by the bacteria, which is very interesting if that's true. I also don't know what to say about the huge concentration of ciliates here. And I'm surprised not seeing anything in the middle of conjugation here, or perhaps the... oh no, that's not a pair of conjugating ciliates. That's a pair of ciliates stuck in the... no, three of them, in a few spirochetes. Again, here's a mesh of the bacteria. Now, the ciliates, some ciliates look very similar and are very difficult to differentiate if they are they have no morphological differences. So, for example, if there are two species of ciliates here, I mean the small ciliates that look very alike, we would need genetical data to identify any of them. I don't see any of the ciliates of this species, the large one that's trying to move around the smaller ones, get stuck on... I'm not sure if this is one. Oh yes, that's one. Yep, they're getting stuck in the bacteria, my mistake. No, that's the biofilm, and that was copepod larva, but here is a very large clump of ciliates stuck again for some reason in bacterial colonies. Let's see if we can find something else to see. Again, I'll keep the slides for next time to observe them after they have been around for several days. But let's go and see what we can find. The problem with observing those larvae is that they're always moving. Here's a dead one. What the... What's happening here? Now, this red spot here is the eye spot of the copepod. And here is the head, which it should be, which the eye spot should be, and it has gone here. Now, this is definitely an infected copepod. Those are probably small ciliates, maybe, and they are all over inside the copepod. You can see them here. But this is a large infection. Those two bubbles, or whatever they are, I don't know what they are. The small spheres. Now, a problem with microscopy, as you may have seen, is that any small movements make the slide and the water in the slide tremble a lot and vibrate, which obviously lowers the experience quality, but sometimes it makes it difficult to observe things because they might move, or you might lose them, especially in higher magnification. Sorry. And yeah. Now we are again at 100 times magnification. Here is something that I don't know what it is. It's probably part of the fungus, if I'm honest, but it could be just dead things. The black stuff. Oh, what is that? This, I think, is a paramecium. I hope it is one. Here it is under the biofilm. Fortunately, if there are stuff blocking the light, you cannot see very well. Even if they are transparent, it makes the light bend in a certain way, not exactly bend, diffract in a certain way. Refract, sorry. I'm messing my words here. Refract in a certain way, which basically does not allow you to see much detail, if you can see here. Now, the black stuff here could be anything really, from dead things, dead organisms, to fungus growing in the slide. So if we don't see again the paramecium, I think that the slide doesn't have any more things to offer. Again, I will be keeping the slide. Here it is. For some reason it goes only moves around the biofilm. I think it is definitely a paramecium. I think this is an alive larva. Let me open a bit the condenser. Yes, it is an alive larva, and I think it's stuck in the biofilm. Here you can see the proper place for the eye spot. It is quite a ways away from the position that it was before, and you can see its appendages, legs you could say, not exactly legs. It kind of walks. Now it's a bit obstructed by the biofilm that's above it. The problem with the biofilm is that it generally floats, so things can easily go under it, but not necessarily over it. And the things under the biofilm are very hard to observe, and things... Oh, I think I can just about see some individual cells. Or maybe not, but I'm not sure. Don't think you can see it. I'm not sure what the spherical thing in the middle of the copepod is. Anyway, let's move on to the next slide, to the next specimen. Again, I'll be storing them for later observation. I think I'm going to look at we're going to look at another slide, and then I'm going to call the stream off because it's getting late here. Let's see. I have this vial which I would like to put under the microscope. It has some algae on. Come on, focus here. Let me... okay. So, again, pull out the slide and put a little bit of solid stuff. You need them for microorganisms to latch on and have a place to again stay, yeah, feed from. I think we will need a thinner pair of tweezers. So there is a bit of algae in the bottom, you can see, and that's what I want to take from. let's change the lighting a bit. There we go, so I can see better. There we go. I just pulled some algae. Oh. Also, it is good to spread the solid material around the slide because it gets less obstructive, and also the slide gets thinner that way. Let me pull a bit of water from a new pipette, because generally I like to use new pipettes for every specimen, because they the specimens have different bacteria and different microorganisms, and the others might disrupt the small ecosystem that has formed there. I think that's enough water. Let's close the vial, put the water back. Close the vial and put the cover slip on. Let me find a cleaner one. There we go. Now this slide is very good. It's quite thin. I hope I didn't crush anything though. There's that danger. And also that is another reason why you want solid material inside the slide. Now I can already see some microorganisms roaming around. Not roaring, obviously. That might be what I'm seeing. Oh, here's another spirochete. Where is it? There is it. A bigger one. Let's go down in magnification again. Now that green stuff could be anything. I'm not sure. It could be a euglenoid cyst or something. Here's another flagellate. I'm not sure. I think it is eating. Oh no. I thought it was eating a bacterial string that's right next to it, but it's just right next to it. I don't know the species again. The larger microorganism is the easier is to identify. Yep, they are flagellates, I think at least. Oh, this one died. It just burst. Generally, when a cell dies, its cell will just bursts open and the cell contents empty. Yeah, I think they're flagellates. Here's another species. this one also, I don't know what it is, the one in the middle. And those green things, the green dots dotted around, are microalgae, I think. But let's go near algae and see what they have to offer. Here's the item. Also, you'll notice that the previous slide didn't have any diatoms. That is because it was a biofilm, which generally floats up, while the diatoms are quite heavy and they sink down. You find generally more diatoms on the sediment. Here is a colony of green algae. Or at least I think it could be a cyst of euglenoid. Now, for now I'm looking for something larger and slower, because they're the easiest to observe and the easiest to find things about. The one thing that I haven't, oh, here's something, the one thing that I haven't found yet, and I'm really sad about, is the stentor. They're large, very large. They can reach up to, stentor coeruleus, for example, can reach up to 4 millimeters in size. And also they're quite thick, so you can easily see them with the naked eye. Here's a dead ciliate, I think. It is dead for sure, but I don't know if it's a ciliate. It's probably a ciliate though. And yeah, the stentors are basically trumpet shaped. Imagine, for example, a trumpet, but blue hue, and moving and flexing around. The stentors are filter feeders, meaning they circulate the water around their oral apparatus, and they filter the things they can eat with membranelles, which are cilia fused together to make sieving structures. No, here's another ciliate, which I don't know what it's doing. And here again there are lots of diatoms. It is from the same sample, kind of. So the filamentous algae with the diatoms are everywhere. Wherever there is filamentous algae, there are diatoms. Here's another diatom. Let me a bit better now. Nope. There we go. Here's a piece of filamentous algae with diatoms again. This could be the Diuclina. I'm not sure though. The thing is, in microbiology, is hard to know for sure things, especially when you don't have methods like genetic sequencing or molecular tests, which obviously I do not have. The other thing that I would like to find today, but I don't think I'll find, is Vorticella. It is basically one of my favorite microorganisms. Um, they have their bells. They're bell shaped with a long stalk, and they are filter feeders too. And they are sessile, firstly, and so they're very easy to observe and document. I intend to make a series of videos about them, all about Vorticella. Um, but for now, um, I would like to have a bit more footage to show. Now, this slide seems to be quite disappointing. Another thing is in microscopy is that you don't know what you'll find. You never know what you'll find. And so you can expect to find some things sometimes, but for example, if you see green stuff, you're obviously you will find algae. In sediment, it is very possible that you'll find little worms. This seems like a shell of something, but I don't really know, and I cannot tell because it is completely opaque. And yes, in the sediment you can expect to find worms, nematodes in particular, and also if the water is very shallow, you can expect to find um, um, diatoms. But otherwise, in terms of ciliates or other microorganisms, it is very hard to judge because obviously you cannot see them before you put them under the microscope, except if they are very large, like stentors, which you can obviously see if you have gotten any. Or microanimals like copepods or daphnias, or for example, larger, not large because they're quite small, large worms, annelids, or insect larvae. Those can be quite common in some places too. But yeah, this slide is quite disappointing. It is not as rich as the previous ones. There is a quite high possibility that in a few days there will be stuff because there are, cysts around that you cannot identify unless they get out of cyst mode. You could say that cyst form, get out of the cyst basically, and or maybe there are small versions of larvae essentially, or the equivalent of larvae for the microorganisms. But yeah. Now what? Oh, it is. We're closing to 2 hours, so I won't be changing slides. But I'll keep looking for a bit, and then in about 10 minutes or so, I'll be closing. Yeah, so you can see loads of diatoms. For example, here is an interesting one. Also, in almost every sample, you can expect to find bacteria. They are everywhere, literally everywhere, in your hands, on your face, inside of your stomach, inside of your everywhere, on top of your desk, on your phone. And in water samples, they're especially abundant because more nutrients, um, and yeah, more possibility to find a host if they're parasitic or if they're infectious to, for example, ciliates or animals. For example, if large animals, for example, large mammals are drinking water off of a pond, and there happens to be a bacterial breakout, for example, infectious bacterial for a particular species of animal. For example, let's say deer. A deer that's infected comes into a pond and drinks water, and a bit of saliva, for example, with a few bacteria drops into the water. They multiply there, and then they have more chances to infect hosts. So this is probably a root of some kind, some plant. You can see the cells, the cell borders. The roots obviously do not photosynthesize because they're under the earth. They're in the soil, in the sediment. Here's a ciliate. Quite a disappointing slide, I have to say again. Yep, I don't think there is anything else to see. Um, this is, I think this is the same species of ciliates we saw in the previous slide. So, yeah. I also see that a few other people have joined in the meantime. Let me give Rose. There we go. So, um, I am going to be closing up for today. So, thank you for watching. Go check out my Instagram and Reddit accounts and YouTube, but this is going to YouTube, but so whatever. Thanks for watching. See you in the next time. Goodbye.